Hi, my name is Mullally. I am an undergrad here at the University of Oregon, and this is my research that I've been conducting this summer for the Undergraduate Research Symposium and for Deanna Flores and Samantha Hopkins here at the University of Oregon's Earth Science Department. 2D morphological analysis of squirrel molars and their implications on taxonomy. Before I continue, I first want to thank the Center for Undergraduate Research for awarding me the FYRE grant this summer. It has been an amazing help in helping me continue my research, and it has been just absolutely amazing. I've had a wonderful experience, and here I hope to share with you my findings in this research that I find quite fascinating. Some background before we begin. My research falls under a much larger main project that is being headed by Deanna Flores, a PhD student currently under Samantha Hopkins at the Paleontology Lab here at the University of Oregon. This larger project aims to understand the taxonomy and biodiversity of extinct rodentia in the John Day Formation. One of the main ways that paleontologists aim to identify species by their molars is by looking at the cusps of the molars and comparing certain aspects and dimensions of them to other teeth cusps in order to differentiate species. However, when it comes to rodentia, this family is very well known for their cusps being worn down over time. So unlike most other mammalian families, it is not a very reliable way to identify rodentia by their molars using their cusps. Unfortunately for paleontologists dealing with extinct rodentia, the most common fossil we find with a lot of rodent samples and species is their teeth and their molars mostly, which is highly unfortunate. And in most cases, there has been a misidentification, most likely in a huge number of fossil species and possibly even modern species that were only looked at by their teeth and identified that way. Um, but this main project that Deanna is working on is aiming to find a new method in order to identify rodentia species variation using their molars still. This new method we aim to use is using the outlines of the teeth instead. Though it has been practiced in other papers and other scientists have taken a crack at it, and so we attempted to make our own version of it for rodents more specifically and to test it on the John Day specimens and variants that we have here. So our methods of research included taking photos of the ground squirrel molars, and we took photos of both the modern and fossil samples. We wanted to do this to be able to, one, test this method on the modern samples that we have much more copious amounts of and much easier access to here at the UO collections. And we also went to the OSU collections and they allowed us to take photos of a decent few of their samples there in order to help add more numbers to our research when it came to our modern data. This way we were able to see, one, if the new method we used would actually properly work, since most of the modern specimens, to our knowledge, were properly identified. And then we would use it on the fossil data that we collect from the John Day location, specifically the John Day National Monument over in Eastern Oregon. And we would aim to compare and combine the data that we find in order to properly create graphs to show us if this new method would properly work in differentiating species. So we take photos of both the first molar of the ground squirrels in both the top and bottom, so usually nicknamed the M1 molar, and also the fourth premolar, which is usually at the end of the molar line, if not right before the end, usually nicknamed the P4. We would use this device we have called a dynolight that is a camera and program that we use at the lab, and it was quite effective in taking photos for us to scale so we could eventually go to our next step, which was to take these photos and isolate the outlines of the molars in TPS util and TPS dig. TPS util, we would turn, basically it would turn all the photos that we had taken of a certain type of tooth type, turn it into a TPS file, and then run that TPS file into TPS dig. And in TPS dig, we would have the ability to go and specifically outline creating two landmarks on one on the top and one on the bottom of the molars in a specific formation. And then we would make 64 point outlines around the molar. After we do that through every tooth sample that we have taken photos of, we take those data plots from that TPS dig program that processed those data, and we put it in, we use elliptical FYRE analysis, principal component analysis on FYRE coefficients, and finally linear discriminant analysis using RStudio, using our own code that Deanna had worked on for a while and successfully tested. This code then would take the data that we had collected and turned it into hard to comprehend lists and lists of numbers into an actual comprehensible graph and data that we could properly assume and find new ways to identify these creatures. Our aim was to see proper variation in these graphs between different species and different landmarks to be able to show that distinctly two different species or multiple different species. In most cases, we would assume for misidentified molar fossil species, it would be looking more like a shotgun blast on the graph plot, as it would be a scatter of these points. Some could be easily picked out as a species in different areas with clear dividings. Some could be grouped together, suggesting they might be of the same species and misidentified. But what we found was some pretty interesting data, actually, after continuing this. So here is one of the photos we took. This is this was a sample that we had at the University of Oregon. It is to scale with this bar here. This is sample 8461. Sorry. It is labeled as Spermophilus beldingi, but I believe the proper species identification, because this was an older sample from early 1900s, perhaps late 1800s, I believe, that was in the UL collections, back when Spermophilus was a major species. And since then, it has been divided into multiple different subspecies, and Spermophilus isn't really a valid species anymore. And so the proper name, I believe, for Spermophilus beldingi should be Uricitellus beldingi. So here we have, this is what I mean by the P4 and the M1. This sample had 5 molars along it, as you can just barely see the last molars there. And down here should be the incisors. So we look from this point, this is the P3, and this is the P4, the fourth premolar. And then here we start the first molar. And so we wanted to look at, on both sides, we wanted to look at these specific molars. So we take photos and then isolate each molar in the photo. Sometimes there'd be one molar in a photo, sometimes there would be multiple, depending on the completion of the sample. And we would, from there, be able to properly put it through the program. As you can see, this is a shot of the same specimen, but the bottom jaw, as we had a complete jaw with this one. And so here, there is no P3 on the bottom jaw. So here's the P4, here's the M1. This here is a lower M1 of a Myoscurus bellovanius, and it is a fossil specimen that we got from the John Day area. I believe this is specimen 12638, And so this is the lower M1. Of course, we don't have a full jaw to go with it, so we had to get creative when trying to find its proper orientation in post-edit when we were putting it through the program, as we had to orient them all in the same position for the program to properly work. So using that, we can see the cusps here just barely, looking like sort of as if you were looking at a plains and seeing the rise of a mountain. This, you can see here's a cusp here, and a cusp here. This is an upper M1 that we had of Protoscurus menyi, sample 4527. Is a bit upside down. You see, this one's harder to find, but you can see there's a cusp here, sort of a cusp here, a bit here too. This is a very clear sample that we got. It was very helpful in the program. Some samples were not as complete or were dirty in a way and did not actually go through the program very well. Some were incomplete. Some were misidentified in their which formation they were. So definitely the research we conducted had a decent few errors that we plan to, moving forward, fix and add more methods to. Now we get to the actual data, the graphs that we found. So this is the modern samples that we have, and is the lower first molar. And so we have multiple different species here. We have Calospermophilus, Otospermophilus, a unknown Spermophilus species. We have Terrius and Uricetillus. And so you can see it looks at first like, as I said before, a shotgun blast. But you can see with the more common species we have a much more big barrier. So you see there's more of these purple ones here. I believe this is Uricetellus, all these little purple stars. This one here, it's like the blue squares with the X's in them. I believe that is Tamias. And you can see there's definite variation in these two. And if we squint and find the less popular species in this data set, you can see actually among the Tamias townsendi, there's also Tamias amurus, I believe that is, with it. So of course they are both different subspecies, but still very closely related, as shown by this graph. Over here we have the lower fourth premolar graph, and it kind of shows the same thing exactly, like and kind of like flipped over as it is the fourth premolar, which is very fascinating. But it shows pretty much the exact same thing as over here. Lower first molar graph shows a definite variation, at the same time a similarity between two closely related species. Same thing over here. Next was of the other modern species, we have the upper first molar graph, which definitely we had less data on, but definitely was a lot more closely together. We assume this might be from errors in our methods and from the process. This one here is more similar to the ones from the previous page. This is the upper fourth molar, or upper fourth premolar of the modern species, and it definitely shows the same thing where there's definite separation in species. Now we get to the fossil species. We had a lot less fossil specimens than modern specimens, to note here, and it definitely shows that there's way more points than there are on the previous graphs. So we can see, though, this is the lower first molars, and there's definite variation. There's groupings. Some of them are more wide because there's more of them compared to other specimen samples here, but you can definitely see there is separation nonetheless, though hard to pick out. Here we have the lower fourth premolar graph of the fossils, definitely even less samples, but still even very much variation, much more so than the last graph actually. This is showing more what we want to see, or more what we are looking for and seeing if is possible. This is the upper first molar graph, which also, again, way less data and way less samples than we wish, but this is what we have to work with at the moment, and we hope to gain more samples in the future to add to this. And you can see there is still, again, definite variation. Now, before I move on, I should note we did not include a graph for the upper fourth premolar because there was and I mean, this looks like very few specimens compared to the modern. The fourth premolar on the upper fourth premolar had even less. It had perhaps 2 or 3 specimens at most. And so we decided against using that fossil data and only used these 3 as they were the most complete. Next steps in our research, we had a lot of inconclusive data because of lack of more specimens, and also because we were using only the outlines, and mainly we wanted to see if it could possibly work. And as far as we were aware, looking at those graphs before and other data we were given, it does work to an extent. So our next steps moving forward, we want to first gain more samples. And also we want to find a way to use cusp data as well and outline data at the same time and merge them together to be able to properly identify rodentia species in more ways than one. So perhaps, though, again, cusp data is, as I said before, very unreliable with rodentia. It still serves its purpose and has identified at least a decent few species correctly. So we aim to now merge that with our new outline method that we have been developing in order to properly find a effective method that we can use to identify multiple different rodentia species. And that is it. That is the research I conducted on specifically ground squirrels. Again, this is a much larger research project being led by Deanna Flores here at the University of Oregon under Samantha Hopkins. Though her main project aims to look at a most wide range of rodentia, my research was mainly focused on the Squirridae family and samples that we had. We would like to thank Dr. Nick Famoso and the John Day Fossil Beds National Monument for providing fossil samples, as well as Alvaro Cortez at the OSU Mammal Collections for providing more diverse modern specimens. We want to thank Miles Hodge for data collection and analysis, and we want to thank Edward Davis and the Museum of Natural and Cultural History for collection samples and resources. And finally, I want to thank the University of Oregon's FYRE program for funding and the Center for Undergraduate Research for the funding I was given this summer. It was an amazing help and definitely made it possible for me to properly do this research. Thank you for listening. I hope you all have a wonderful day. Goodbye.